Good morning, I am Monica Ramchandani. Today here to present my research on fruit leaf disease detection using lightweight deep learning. So we'll go through introduction, literature survey, the dataset which we have used, the methods which we have used like CNN, activation function, ReLU, ADAM, results, etc. And finally the conclusion and the references. So India is one of the world's largest fruit growers, and there is also horticulture production in India, and plants need to produce high yield to fulfill the increasing demand with the growing population. So various strategies are required for protecting the plants from the insects and the pests, and detecting illness in fruits is crucial to determine the yield of crops. Banana, it is one of the world's most widely consumed fruits, and grape is a fruit which is very widely used in making jams, grape juices, etc. So we have gone through a few papers. So before we discuss our method, let's summarize these papers. First is deep learning precision farming, grapes and mango leaf disease detection by transfer learning. They identified the diseases or their absence. Deep CNN, AlexNet were the methods which were used, and 99% accuracy was observed for grapes and 89% was observed for mangoes. And citrus fruit disease detection and classification using end-to-end anchor-based deep learning model. They classified most likely target area, and they made use of two-stage deep convolution neural networks, and 94.37% of accuracy was observed with an average precision of 95.8%. A fruit disease diagnosis model based on stacking and ensemble learning, validated and tested against 5 models, ensemble and deep learning. An accuracy of 98.05% was observed on a validation dataset and 97.34% on the test dataset. Guava disease detection using deep convolution neural networks, a case study of guava plants. Deep convolution neural network, they used color histogram equalization, and AlexNet, SqueezeNet, GoogleNet, ResNet 50 and ResNet 101 were the methods used, and 97.74% of accuracy was observed. So the dataset we have used is from Kaggle, and it is banana leaf disease and grape leaf disease dataset. For each fruit we have detected 3 illnesses. For bananas, the illness is bacterial wilt. In the first picture you can see it. Next is sigatoka, then cordana, and for grapes it is black rot, esca and leaf blight. We have also took the images of healthy leaf to classify it as a diseased leaf, and a healthy leaf and diseased leaf consists of 3 more diseases. So we have classified our data as training data, validation data and testing data, and the images of these are given as an input, and the images are the methods are applied to it to get the final result. So we have used the first method we applied was lightweight convolution neural network. Lightweight convolution neural network, it is a feed-forward neural network which will evaluate the visual images by processing the data and in grid-like layout, and a convolution neural network is used to recognize the things in an image. So CNN will learn these filters and characteristics, and they are classified into 3 strata. One is convolution layer, pooling layer, fully connected layer, and in addition to these, 2 more layers are used in a 5-layer convolution neural network. Those are dropout and activation functions. So convolution layer, it is the first step process of extracting the valuable features from an image, and the convolution layer has several filters to perform this convolution operation, and every image is considered as a matrix or pixel. Next layer is pooling layer. Pooling layer is a downsampling operation that reduces the dimensionality of the feature map. It converts it into a 2-dimensional feature map or rectified feature map, which goes through the pooling layer to generate a pooled feature map, and it uses various filters to identify the different parts of the different images, like the edges or the corners or the body parts or whatever. And the next step in this is flattening. Flattening is used to convert the resultant 2-dimensional array from the pooled feature map into a single long continuous linear vector. So this flattened matrix, it is fed as an input to the fully connected layer to classify the image. And we will discuss about activation functions further. As you can see in the picture, the flattened matrix is given as an input to the fully connected layer. There is a pooling layer, and the image is given as an input to the convolution layer, which is again pulled into a feature map using the layers. Next is activation function. Activation functions are used to determine if the output of a neural network is a yes or a no. It will transform the numbers from -1 to 1, 0 to 1, and so on. And there are generally two categories of activation functions: linear and non-linear activation functions. Linear activation functions will allow themselves to adjust them, and non-linear activation functions are used to categorize them. And examples of non-linear activation functions are sigmoidal or logistic functions, tanh functions, ReLU and leaky ReLU. So sigmoidal activation function, it is also known as logistic function, and it is a function which has an S-shaped curve, and it is generally used where the probability prediction is the output. And the range is 0 to 1. Because probability is predicted in the range 0 to 1, it is used for this, and it has an S-shaped curve, and this is the formula for it. And tanh function, it is also called as hyperbolic activation function, and it is related to logistic sigmoidal function. It is similar to it because it has an S-shaped curve even, but the range here is -1 to 1, so it is more advanced than that. And the function is monotonic, but its derivative is not monotonic, and it is used to distinguish between two different classes. And next is ReLU. ReLU is rectified linear unit. It is generally used for once the feature map is extracted, the next step is to move to a ReLU layer. So what does ReLU do? It will perform element wise operation and sets all the negative pixels to 0. And it is represented by the ReLU function. F of x is equal to max of 0 comma x, and it is the most used technique. And the advantage is that it does not produce vanishing gradient problem, as the number of layers will grow. Next is ADAM. ADAM is adaptive learning rate approach, which will calculate the rate for individual learning defined on various factors of learning. It is a combination of RMS pop and stochastic gradient descent with a momentum. It uses the squared gradients to scale the learning rate like RMS pop, and it takes advantage of the momentum by making use of average of the gradient instead of gradient itself. And it is an adaptive learning rate method, and it is an Adagrad algorithm, and it uses the below formula, and it computes individual learning rates for different parameters, and its name is derived from adaptive moment estimation. And that is why it is, because ADAM will use the estimation for the first and second moments to gradient to adapt the learning rate for the weight of the neural network. And so after applying these algorithms of optimization and classification, we got the two confusion matrices. One is for banana towards the left with the 3 diseases and the healthy leaf, and second is for the grapes with the diseases and the healthy leaf. And we have obtained an accuracy of more than 80% and more than 90% for grapes, as you can see in the graphs. So I'll show you this is our code for the grape disease, and here the layers are applied, and we obtained the following graphs for accuracy, and the following confusion matrix for the grape. And we have observed as the number of epochs will increase, the accuracy will increase. So presently we have applied 15 epochs here. We have also done it with 25 epochs and 50 epochs also, and the same is for the banana also. And so this is the paper, which contains abstract, the keywords, the introduction, and the literature. We have done it for more than 10 papers, maybe 15. And this is the methodology. So this is the architecture of our model, which where the crops depend upon the temperature, soil, humidity, and the type of soil which is there. And then the farmer can access it through a cloud, and we can determine it. So the diseases which we have predicted, and next is cloud computing. Then the method which we have used, CNN and its layers. And these are the formulas which are for the pooling layer and the fully connected layer, convolution layer, and the activation function, which is linear or non-linear, and the ReLU, rectified linear unit, and ADAM, this is an Adagrad algorithm, and finally the results and conclusion. So finally I would also like to conclude that in this we have predicted the diseases, 3 diseases each for banana and grapefruits, and we have applied some deep learning, lightweight deep learning based models like lightweight CNN and its activation functions like ReLU, and we have also applied the deep learning based optimizer, which is Adam, and the prediction of accuracy was more than 80% as I showed, and as the number of epochs increased, accuracy was also increasing. So these are the references, and thank you.